First-in-human study, iPSC-derived CD19/BCMA CAR-NK therapy reverses fibrosis in systemic sclerosis. In June 2025, something remarkable happened in the world of autoimmune medicine. For the first time, researchers reported the successful use of an allogeneic iPSC-derived dual CAR-NK cell therapy called QN139B in a patient with severe, treatment-resistant systemic sclerosis. This isn't just another incremental step, it's a potential paradigm shift for diseases where effective therapies have been frustratingly out of reach. So, what exactly is QN139B? At its core, QN139B is a natural killer cell product, but it's not from a donor or the patient. Instead, it's engineered from induced pluripotent stem cells, or iPSCs. That means it's off the shelf, ready to use, and doesn't require the time-consuming process of patient-specific engineering. Even more impressive, it targets two key molecules, CD19 and BCMA. This allows the therapy to eliminate both B cells and those long-lived plasma cells that drive autoimmunity and fibrosis in systemic sclerosis. What about safety? The cells are designed to minimize alloreactivity, so the risk of graft versus host disease is low. Plus, thorough genomic screening confirmed no chromosomal abnormalities. In the first treated patient, there were no serious toxicities, a result at least as favorable as what we've seen with CAR-T therapies in autoimmune settings. Now, let's talk results. After a single infusion, the patient experienced profound depletion of B cells and plasma cells without major side effects. Over 6 months, there were clear clinical improvements. Skin and organ fibrosis regressed, and histopathology showed restoration of normal skin and microvasculature. Even the autoantibody titers fell, hinting that the underlying autoimmune process was being reversed, not just suppressed. Digging deeper, single-cell and proteomic analyses showed a shift toward naive B-cell phenotypes essentially, a reprogramming of the immune system. There was targeted elimination of pathogenic B cells, and levels of pro-inflammatory, pro-fibrotic proteins dropped significantly. Why does this matter? Systemic sclerosis has long been a therapeutic challenge, especially once fibrosis sets in. By specifically targeting autoreactive B and plasma cells, QN139B offers a precise, durable, and scalable immune reset. This approach could open the door not just for scleroderma, but for other severe autoimmune diseases like lupus or myositis. Of course, this is just the beginning. More trials are needed. But this first-in-human success shows that off-the-shelf iPSC-derived CAR-NK therapies may finally offer a safe, effective, and accessible option for patients facing the toughest autoimmune diseases. Stay tuned. This is just the start of what could be a new era in immune modulation.